What is fermentation? This is where the yeast come in and they eat all the sugar, they produce CO2, and they produce alcohol. That's the byproduct of it. They're making the beer. So they're eating all this sweet liquid and they're turning it into alcohol and CO2. Now after that's done, the yeast flocculates out, which means it basically comes out of suspension and goes to the bottom. When it's at the bottom is when we crop it off. We either harvest it or we get rid of it. Usually it's really good for B12, riboflavin. It's good for cows. Gives them a lot of flatulence, makes them a lot of methane and stuff. But it's good for you. There's nothing wrong with having a little bit of probiotics there. And then after, so you've taken off your yeast, now we'll put it into cold conditioning. Basically what we do is we turn on a jacket. There's a jacket inside of this that runs glycol, cold glycol back and forth. As it's running back and forth, it'll cool that on the inside. This is where the beer matures. Prior to this, it's green. But after it gets cold for a certain period of time is when it matures. When it matures, it actually has a nice flavor of beer. Before that, you're going to think of like green apples or acetaldehyde or some other type of green flavor that you're not really going to like. The beer's not done yet. The beer's green. The beer's immature. You want to let it mature. It'll mature for about 2 to 3 weeks in here before the flavors really become very nice and round and easy and supple. With lagers, it's anywhere between 6 and 8 weeks, so it can be a lot longer. It just takes longer. It's slower. It's just the yeast function of the yeast and temperature. After that time, we will take that beer. Now it's finished beer. All the sugar's gone in it that we want to have gone. All the alcohol's in it that we want to have in it. And we put it into a bright tank. Another sanitary connection, another sanitary hose, another sanitary tank. I've been through all the chemicals and everything else, and then it's been purged with CO2 or nitrogen so that it doesn't get oxidized, and then we put it from one tank to the next under pressure. This is where filtering will occur if we filter. We don't necessarily need to filter. If you let the beer set long enough, the yeast will flocculate out. If it flocculates out, the beer turns bright. If it's a little bit hazy, I'm okay with that. That just says that all the goodness is still there. So now, once it's in the bright tank is when I force carbonate it. I've got a carbonation stone in there, much like a fish tank. A stone goes in there and it pressurizes it, diffuses gas, and it pressurizes that liquid, and it brings up the volume of CO2 in there so it has a little bit of a sparkle, a little bit of a tickle, a little bit of a burn on your nose. This is a good carbonation of beer. Or we can do this with nitrogen, and then you nitrogenate the beer.
Yeast is what actually creates the beer. The brewers will actually create the wort, and then they in turn will pitch yeast into the fermenter so that the yeast can then turn the beer into alcohol and other byproducts as well. Yeast is very important to fermentation because without it we wouldn't have beer, and it also helps sterilize the beer as well with some of the byproducts it creates, like the light amount of tetracycline.
Yeast affects the taste of beer. The best way that I know how to explain it, it's like you. Each one of you are a yeast, a different yeast culture. You all smell differently. Everybody could wear the same cologne, but you would still smell differently. And so yeast cultures are kind of that way. They all smell differently. So what we try to do, lack of a better term, is to clone these yeasts. We don't really clone them, we just grow them up. If they're all exactly the same, they're all going to be exactly the same. It's kind of like going to your supermarket and picking out all the asparagus. It's all going to look the same. It's all varietal. It's all going to be the same. So yeast give it the flavor of the beer. They will also give you the alcohol potential for the beer. Some yeast love being at the buffet, at that sugar buffet, and eating all day long and just producing all kinds of alcohol. And they're just fat, sluggish yeast cells, and they'll just consume alcohol, and they'll consume sugar, and then they'll just pee all this alcohol. And they're very tough, and they can be out there and they can survive that alcohol content. Most brewers' yeast aren't that way. They're little wussies. They go to the buffet, they get their little bit, they make a bunch of babies, they eat some, and then they kind of go to sleep. That's pretty much what brewers' yeast do. They kind of hang out, make a bunch of babies, and then kind of go back to sleep. Their alcohol tolerance isn't great. Once it gets up to about 7-8%, you start getting some mutations. When you get mutations, you get flavor components that taste different in the beer. Some are preferred, some are not preferred. Some are like bubble gum, some are like clove. All depends on the style of beer, and the style of yeast or the varietal of yeast that you're using will definitely make a different flavor of the beer and different aroma of the beer, and also potential alcohol. Yeast doesn't give you the alcohol, it's the amount of sugar that you put in there. Imagine making a cup of coffee and you make it twice as strong. You use twice as much grounds, half as much water, and your coffee is going to be twice as strong. It's the same thing here. If you use twice as much grain, half as much water, you're going to get a stronger, more viscous, sugary liquid, so thus your potential for alcohol is higher. You're still going to pitch it with that yeast, but depending on that yeast culture, whether or not it's going to be alcohol tolerant. Okay, so there are two basic yeasts that we use here in a brewing environment. It's an ale yeast and a lager yeast. There's Saccharomyces cerevisiae, which is your ale yeast, and your Saccharomyces, which now has been reclassified as Pasteuranus, which is the old Carlsberg yeast. And so there's lagers, which is the Pasteuranus, and then there is the Cerevisiae, which is the ale. Both of them are Saccharomyces. The big difference in these is, I always tell people, do you like working outside in the heat, or do you like working inside the office and at a computer desk? Because that's the big difference. If you're an outside person, you like the heat, you're an ale yeast. If you're a lager, you like working inside in a cold, air-conditioned environment, probably at the computer. Yes, you're still just as active, you're getting things done, but you don't put up a big sweat. You don't really hustle around and do things. You're a little bit slower. So with lager fermentation, it's colder. 45, 54 degrees Fahrenheit, anywhere between 9 and 10 degrees Celsius. So it's a colder, longer fermentation. And there's kind of a misnomer that most of the fermentation happens at the bottom. The yeast culture for lagers do like going to the bottom of the fermenter, but they're all up in the wort anyways. They're fermenting up in the beer. And same thing with ales. What happened was, way back when, is they used to top crop yeast. What that meant was is they had an open fermenter, and as the yeast was bubbling up and over in Kreuzen, they would come with a stick and they would crop off that yeast, dump it out, and then they would get another stick and they would crop that yeast and put it into a bucket. They just knew that it was something that you had to have something for another beer. They didn't know exactly what it was. They just knew it was something you had to have. They put it into a bucket, and then for their next batch of beer, they would use it again. For lagers, they wound up being able to grab it from the bottom. So this was a big invention. Once they were able to grab it from the bottom, they were able to grab those lager yeasts. So now today we have several different varieties of lager yeast and several different varieties of ale yeast. Lagers normally are colder, slower fermentation, higher risk of DMS and diacetyl, but well worth it because it's very clean and crisp, and there's nothing like a good lager. Ales are very fruity and estery and wild and kind of, yeah, they're kind of like that. They've got some funk maybe sometimes, but that's what they are. They're more fruity and estery. They're that person that went to the gym and they started sweating and you can smell their cologne throughout the entire place.
One of the biggest importance of a microbiologist in a brewery is to ensure not only the quality and the taste of the beer, but if there are any sort of biological contaminants, to be able to identify where and who may have contaminated the beer. Some of my common duties is to check the Play-Dohs of the beer, as well as the temperature, to ensure that everything is going smoothly during the whole entire fermentation process. Other duties can also include checking the yeast viability as well as the yeast count, especially if we're going to be reusing the yeast for a future batch. As a microbiologist, one of the biggest hazards for the microbiologists themselves is probably going to be when they're working with alcohol or any type of flame. One of the most common errors that a microbiologist can help in a brewing could be whether or not they are pitching the correct amount of yeast into the fermentation tanks. If you're either over pitching or under pitching, it can actually cause undesirable taste in the fermentation process. So one of the pros and cons of yeast harvesting could be whether or not the microbiologists themselves harvest enough yeast that can be used in a future batch. It kind of helps to work with the head brewmaster to figure out how much yeast is going to be needed for a future batch. And this can be simply done just by doing some future planning, as well as counting some of the yeast cells in order to determine what is the viability of the yeast cells, as well as what is the total cell counts per ml of the yeast. Once this is accomplished, you can then determine how much liters you need, or if you are capable of doing so, you can also get a brink started as well. And what a brink will do, it'll actually harvest off all the yeast in that fermentation tank so that some of it or all of it can be used for a future generation.
One important tool in a brewery is the microscope, but before one can begin using a microscope, one should familiarize himself with the various components that make up the microscope. These are the eyepiece, which one looks into to view what's being on the stage. The rotating nose piece, which actually holds the objective lenses. The objective lenses themselves, as well as one should also be able to identify what is the magnification of each lens. The stage, the condenser, the stage adjustment knobs, which are located on this side right here, the fine and coarse adjustment knobs, as well as the base of the microscope and the arm of the microscope. So the purpose of a forced wort tube is to determine if the wort coming through the heat exchanger is contaminated with anything, including bacterial or other wild yeast, okay? Typically the heat exchanger is cleaned prior to its use for transfer and cooling of the wort from the brew kettle. However, it's still necessary even after cleaning to take a test just to see if there's any residual contaminants left over. A forced wort is very simple. Once you aseptically take a sample from the heat exchanger, you just leave it in the incubator at 30 degrees Celsius, 29 to 30 degrees Celsius, to incubate for 2 to 7 days, okay? You can actually read the samples periodically and leave them there so long as it's done aseptically. Typically after 2 days, you can see whether or not your wort is contaminated just by looking at these two examples here, okay? This one is going to be a little bit more turbid, which is indicative that there might be some contaminants in it. However, further testing is needed, especially when working with already hazy beers, such as a hazy IPA. However, this one is clearly positive, and the way that can be seen is how transparent the beer is, or how clear the beer is here, or the wort. Okay. One important technique that a lab technician or a cellarman can do to determine whether their beer is fermenting correctly is by doing a wet mount. Now, the purpose of a wet mount is to view the health of the yeast during the fermentation process, as well as various samples. In order to begin, you're going to need a glass slide, and it can either be frosted or not, cover slips. Depending on the situation, you may need DI water or not, a transfer pipette, or your sample directly taken from the fermenter. In this wet mount, I'm going to be showing you how to create a wet mount using a sample directly from the fermenter. Before beginning, one does need to sanitize the entire countertop with 100% isopropyl alcohol or ethanol as well. If your lab does provide germicide solution, you can use that as well. You will also need a flame just in order to maintain an aseptic working place. And once all of that is completed, you will also need a flaming lube or an inoculating lube. To begin, you will take your glass slide and your transfer pipette and take a sample from your collecting tube. You don't need much to work with, but just about a pea-sized drop should be enough to be placed on the glass slide. Once the drop has been placed on the glass slide, you then will take one cover slip and place it onto your glass slide. Now, it's important not to put it directly on top of the glass slide, because otherwise you'll create a lot of air bubbles. So when you place it, you want to put it at an angle of roughly 45 degrees and place it down gently. The more gentle, the better. Depending on your sample, you're still going to get some gas bubbles simply because of the fermentation process. Carbon dioxide is produced, but it'll actually minimize the amount of bubbles we'll see here, okay? Once done, you may then turn off your flame, and you now created a wet mount, which will then be placed underneath this microscope for viewing. All right. Once you finish making your wet mount, the next step is to place it onto your stage. Make sure your stage is reset all the way to the bottom. And then just clip your slide in. Make sure it's properly secured. And make sure you start with the lowest objective setting. In this case, the objective setting of this lens is going to be at 4x. Turn on the power source and use the coarse adjustment knob in order to properly focus into your sample. Once properly focused, you can then switch to a higher objective or move back. Typically, oil immersion is not necessary when viewing yeast cells. So the second way of making a wet mount is by taking it from a plate. So after you're done plating some of your samples, typically from the fermenter or any other various sources you can find at the brewery, you then must identify to see if what is growing on the plate is yeast. If you have plates that are using cyclohexamide, that will actually inhibit the growth of eukaryotic cells, including yeast. But in some labs, working with cyclohexamide can be dangerous, so they do use plates without cyclohexamide, but however, yeast can grow on it. So in order to identify if this growth is yeast or bacterial, we must create a wet mount. Similarly to how we did the wet mount when taken from the fermenter sample, we are going to be placing a drop of water this time onto the glass slide, and again, no more than the size of a pea. Then we're gonna heat up our inoculating loop until it is red hot. Okay. You want to make sure that the entire length of the loop is decontaminated by making it turn red hot all the way through. Once this is done, make sure that your loop does cool, otherwise you'll burn the sample on the plate. Typically this is around 20 seconds worth of waiting. One way you could do it is by singing either ABC or Mary Had a Little Lamb and then wait until it cools off. Okay. Once it has cooled, you want to take the least amount of sample possible, otherwise you'll overcrowd the amount of water and space you have on your wet mount. So just a little dab. And then just mix it onto your drop of water. Decontaminate your loop again by flaming it over the flame until it is red hot. Once that's done, you can set that aside. And then again, place a cover slip over it at approximately 45 degrees to avoid air bubbles from forming. Our new wet mount is now ready to view underneath the microscope as previously shown. The forced diacetyl test is a very important sensory test in order to determine if the fermentation has truly been completed in the fermenting vessels. In order to perform the forced diacetyl test, you'll need a flask, some sort of rubber stopper, and the most important thing will be a thermometer. Okay. Once you have all of these components put together, you can either use a stir plate, a microwave, or even some sort of flame source in order to heat up your sample to approximately 63 degrees to 65 degrees Celsius. So what we will be doing is turning on our heating plate, stir plate, and allowing our sample to heat up. Usually this entire process can take up to 20 minutes on a stir plate. If you use something like a microwave or a flaming source, it would actually be a lot quicker. But all you need to do is monitor it. Do not leave this unattended, otherwise things can happen, and you definitely don't want finished beer products spilled all over your laboratory. Just periodically check the temperature by slowly lowering your thermometer or your temperature probe and just reading it as such. Once it gets to approximately 63 degrees or higher Celsius, you then will cover it and then leave it at room temperature to cool for about 30 minutes. And all you need to do is come back, open the tube, open the flask, and then just smell. If you smell something like buttery popcorn, then you know that you need to stop from harvesting yeast in your fermenting vessels so that it can finish up taking up all the byproducts so that you will not have any diacetyl residues in your fermenting vessels. One important step for a brewer to brew consistent beer is to know how much yeast they're pitching every time. In order to count the cells of yeast or the density of yeast per ml, you're gonna need 3 flasks you're gonna need your stock yeast cells, which is typically taken when the yeast is harvested at the time that it is harvested. 2 flasks filled with 90 mLs of DI water. 2 pipette tips, preferably those that can go up to 10 mLs. And your hemocytometer with its appropriate glass cover slip. Other things you will need are a pipetter and 1% methylene blue. So the first step in counting your yeast is once you finally have your yeast sample, which is your stock solution slurry, you're gonna agitate it for about 20 seconds. And this is just so that you can get any of the yeast that has settled out to resuspend into the solution. You never want to take a sample when the yeast is settled out, just because it can give inconsistencies in your counts. Okay. Once that has been done, you will then open up your pipetter, install it as so. Never put your hand in front of it because there's always a chance that it can break and you can actually cut yourself or wound yourself. And then you're going to measure the appropriate amount, which is going to be 10 mLs. For these particular dilutions, we are actually going to be making a 1 to 10 dilution, simply because it makes the math much easier and I get more consistent results that way. Once you finally pour the yeast into your 1 to 10 dilution, which is labeled right here in white, you're also going to plunge everything back down, redraw some of the solution, and allow it to move down again. The reason for this is because there are yeast cells found on the side of this, and since we actually drew that up initially, we want to make sure that they do get washed so that they can come off the wall of the pipette and you can get a more representative result of what is actually found in the stock solution. So that's number 2 and number 3. You're then going to discard this off to the side, and then you're going to agitate this as well. I do have a stir rod in here, which we're going to be using later for when we add methylene blue, but for now, you can just agitate as so. Again, for 20 seconds. I should also note, be careful not to spill anything, because it could cause deviations in your counts. Once you have thoroughly mixed it, make sure to gently agitate it again before you draw some up. And again, you want to take 10 mLs. Now, I failed to mention this previously, but when you take your measurement, you want to draw up a little bit past the 0 mark here. Since this is actually from 0 to 10, it actually counts it backwards. However, if you turn it to the side, you can also see that it goes from 10 to 0, just to let you know how much volume was drawn up. You want to go a little bit past the mark, just because when you let it go, the buoyant force will stop acting on the pipette, and it actually will go a little bit lower. So you can actually measure where the meniscus line is. So, not sure if the camera can pick that up. Perfect, right there. Once that's drawn up, you then want to again carefully place it into the next one so that you can prepare your 1 to 100 dilution. Typically on an industrial scale, you will be using the 1 to 100 dilution because it doesn't overcrowd the hemocytometer. You want your counts to be anywhere from 100 to 50 cells per field, so that way you can ensure the accuracy of it. The hemocytometers are accurate within a certain range. If the numbers fall below or above that, you actually are increasing the amount of error in your calculations. Once it's done, we set it to the side. And to our 2 samples, the next thing we're gonna do is we're gonna add methylene blue. In most cases, you're gonna add about like 1 ml into each flask. But because our 0.01 methylene blue is very effective, we're actually gonna add just a couple of drops and we're gonna eyeball it for this case until it is around the color of mouthwash. And then we're gonna mix it up. And that does look about the color of mouthwash. This one's more so than the other one. And we're gonna wait about 1 minute to let the dye absorb into the non-viable cells. So we're gonna pretend to wait for a minute and then come back to this one right here. So once your solution has settled for about 1 minute, you're then gonna gently agitate it again, just because we don't want any yeast settling out. We're gonna grab our transfer pipette and we're gonna just draw up just a little bit of the sample to place on our hemocytometer. When looking at our hemocytometer, I actually have the glass cover slip on top. That's how we're gonna be adding it. You typically use a micro pipette and load about 10 microliters. I've seen some people even load up to 100 microliters just to fill all the wells on the inside. But for this case, you're just gonna use a pea-sized drop right at the edge of the cover slip, and capillary action will actually absorb that drop. So even if you miss it a little bit, do not worry, it'll actually take it in eventually. Depending on how clean your slide is, it'll do it faster or slowly, or maybe not at all. If it's not working out, you're gonna need to wash it under DI water, and then just clean it with Kim wipes and 100% isopropyl alcohol or ethanol, whichever comes first, okay? Always make sure that your slides are clean so that it doesn't distort any of the images that you may see underneath. But for this case, this one's perfect and ready to go. We're just gonna mix it up again, a couple of seconds, draw some up, and then just place one little drop on here. And again, I can actually see the capillary action and the drop completely disappeared. And you're gonna do it for both sides. A little bit more. Now, if all works well, under the 10x magnification, we should be able to see even dispersion of the cells within all the squares. For this particular case, we're gonna be using the center grid, which contains 25 smaller squares at the center of the hemocytometer. Your hemocytometer actually has two viewing fields, one on each side, that are split in half. So be sure to view both of them to make sure that they are both representative of each other. If they, for some reason, look like one has more cells than the other, you should clean it and restart your entire process just to be on the safe side to ensure that you are being accurate with your results, okay?
So when it comes to yeast harvesting, one of the great benefits about it could be that you will be saving money in the long run by repitching yeast after it's been collected from a fermenter. Typically, this is done when fermentation has ended or has come to a halt in order to catch the yeast during its greatest viability. One of the downsides of actually reusing yeast is the fact that you can actually instill off flavors into the new batch of beer simply because the yeast have been used for quite some time and they can actually be mutated after that. Typically, yeast is harvested around the end of the fermentation phase so that it can be used for another future brew. So what you're going to need is several pieces of equipment that have already been pre-sterilized before the process has been made. And this will include a collection bottle right here, which has already been sterilized with iodine and still filled with the iodine right now, as well as a collection hose, a clamp, the appropriate gaskets, as well as a hose adapter. So the first step is to sterilize the port. Remember, you want to keep this entire process sterilized. And for this, you only need 50% alcohol, which is gonna be found in the spray bottle right here. Next is assembling your collection hose, which is just made of rubber tubing. And so you insert the small gasket into the inside, screw on the hose adapter. And again, we want to keep everything clean and sterile, so more alcohol, as well as adding the gasket, and then carefully placing the hose onto the end of this port right here. So just for safekeeping, we also like to add more alcohol into the hose. And it doesn't really matter because we won't be collecting this, since the first 1,000 mLs is going to be poured straight into the drain, since that's kind of like our runoff, where like just mainly dead yeast is going to be found during that first collection of the liter. Okay. So once you have your apparatus completed, the next step comes in emptying out your bottles, and this can go straight down the drain, no problem. We're going to be dumping the first 1,000 mLs of the yeast simply because that's where basically all the cells have fallen down to. So we really don't want to collect that because it can also skew our numbers for when we're trying to take the viability of our yeast counts, which we'll be doing later today. Our yeast is moving slowly. It's a little bit clogged and jammed just from all the dead cell buildup. We actually have to add CO2 into the fermenter in order to increase the pressure in the top so that we can force the bottom of the yeast to come out. So what we're gonna be doing is we're gonna be connecting this hose right here, which is gonna be filled with CO2. And again, we're gonna sterilize everything, as well as the end of this adapter right here. Attach it, and then open up the valve, the butterfly valve right here on the side, and then turn on the air. You don't need a lot of pressure in order to force the yeast out. Anything less than 5 PSI is good enough. Anything more than that, and then you start risking increasing too much pressure and splattering the yeast all over yourself. So after we added CO2 and we've carved up our fermenter just so that we can make it easier for the yeast to come out of the port, we're then going to be releasing the first 1,000 mLs. And so even with CO2, it can take some time. Just give it like a rough estimate of 1,000 mLs. And if it needs a little bit of motivation, you can also walk the butterfly valve just so that it can further remove some of that backed up yeast into the valve. So that's what we're actually looking for, is for it to speed up, kind of like that. And we want to keep everything sterile because this is going to be going into the collect container. And then we're going to collect up to 900 to 1,000 mLs. And so we just have to be patient and wait for this to collect.